All right, when we look at the large intestine compared to the small intestine and stomach, we do note some difference. And you could probably say that the large intestine looked a little bit more like the stomach than it does the small intestine, honestly, because it is comprised of these pits that you see here. But because there's not any digestive process, the chemical breakdown of anything, we're all done with that. You don't have to worry about any specialized cells. So most of these cells are going to be absorptive cells and goblet cells. So they show you here again the same basic cell structure that we had in the small intestine, but really these cells wouldn't be making any digestive enzymes of any sorts because we're done with that. The mucosa would be here with lymphatic nodules, and then the muscularis would be pretty, which is not, again, here's part of the submucosa here. The muscularis is not quite as well developed, which is one of the reasons why food moves very slowly in the large intestine. So simple columnar epithelium, except at the anal canal, which becomes stratified again. No villi or circular folds, right? Just no enzymes made, lots and lots of mucus when you look at this histologically. You see these structures called the tenae coli. So there are 3 bands of smooth muscle in the longitudinal layer. So the longitudinal layer is significantly reduced. There's a kind of constant contraction that occurs, which results in a puckering we call haustra, shown here. And then there's these epiploic appendages, which we're not really sure what they do, but they're little bits of fat, visceral, fat trapped in the visceral peritoneum, the serous layer that covers all these structures. All right. If we look at the anal canal, we would see an internal and external sphincters. Internal is involuntary, external is voluntary, so difference in the type of muscle. This is pretty much stratified squamous epithelium here, long columns and lots of mucus. So the anal canal is pretty short. The bacterial flora that you find in the large intestines and colon are extremely important to our lives. They are generally, bacteria that enter from our food are generally killed by all these defensive properties we have throughout the tract. But there are about 700 different species, numbering in the hundreds of trillions of cells that live here as not disease-forming symbionts. So they coexist with us. We have an intimate relationship with them. We actually would call it a mutualist. Mutualism is you both benefit. So these bacteria help us with digestion and absorption, and they synthesize a variety of, these are B vitamins and vitamin K. They do not cross the mucosa because of the immune system, so they're actually blocked from doing that. They simply remain here associated with the mucosa of the large intestines. All right? Functions are, again, really just this last bit of absorption, water, electrolytes, and any nutrients that our microflora make for us, we also absorb here. And move it along and really eliminate. So it takes quite a while, because I said we don't have a very well-formed muscularis here. The indigestibles, again, are things that we never evolved enzymes to take apart, break any bonds. So it just so happens that our microflora actually do have the enzymes, and they live off. That's why this is a mutualistic arrangement between us and them. Water and electrolytes. Mechanical digestion is really just mixing the bacteria, exposing them to these indigestibles, and then moving the stuff along. We call it haustral contractions. So that's our mixing. And it's kind of similar to segmentation, but not exactly the same. All right, and then peristalsis is the propulsive. We have these mass movements that happen a couple times a day, and they move the food stuff, which is really mostly, waste product, along quite a distance at a time, about 20 centimeters at a time. I mentioned here that movement is influenced by the presence of food in the stomach. So yes, it's called the gastrocolic reflex, as I said in the last video. And so when food enters the stomach, this actually triggers, with the release of gastrin again, the movement of food through the colon. So again, the stomach directs a movement throughout the tract, is what we've told you now. All right. So any sort of chemical digestion is really the bacteria. So we have this fermentation that occurs, but that is bacterial. That's not your cells. So I'm not taking that back. Your cells do not do chemical digestion in the large intestine. That's all finished up in the small intestine. The absorption of little bits of water, you still have about 100 milliliters you would like to recover. And if we don't recover enough of that, we call it diarrhea because it moves too quickly. If we recover too much of the water, you're constipated, and that again comes from too slow a transit. Electrolytes, sodium chloride, some potassium, right, are pretty much what help move the water. So we have these little bit of electrolytes that get absorbed and water follows them. And then vitamins, we said vitamin K, B12, thiamine, riboflavin are made by the bacteria, our microflora, and when they use, again, our indigestibles. So I mentioned that you can upset your electrolyte balances with diarrhea, and we'll talk about that when we get to the chapter. We'll talk about it again when we get to that chapter. And I said, so for every 500 milliliters of food that enters, about two-thirds of that will be absorbed, and only a third will become feces. All right, defecation reflex is we now need to get rid of it because we said that's a big job for the colon is elimination. So after a couple of these mass movements in a day, food will enter the rectum. It becomes stretched, so stretch receptors send their sensory to the spinal cord. The parasympathetic directs from the spinal cord, that's where the sacral region, directs and sends a message back to the sphincters and to the muscularis, so you get contraction of the rectum, push, and relaxation of the internal anal sphincter. Feces moves into the anal canal, and at that point you'll have sensors that tell you become aware that it's in the anal canal, and generally you will direct then the contraction of the external anal sphincter, right, so that you don't just poop there, all right? So I do mention here, just so you know, you can have as many as 2 or 3 defecations, right, release of feces a day. It can be, it's still acceptable 3 or 4 times a week, but beyond that, if you're not eliminating 3 times a week, there is a problem, and you need to seek help. All right, so the accessory organs. So we've gotten everything through this tract, so what do we have in the tract that's going to help us? What do they make, and how does that help? So the liver is really just does bile. The liver is responsible for a whole lot of things once food enters the body, but in the digestive process, just getting it in, it's bile. All right. So, and I mentioned here again the accessory organs. The small intestine needs, right, what the liver makes, what the pancreas make, all right, to do its job. So 4 lobes, we look at this in the lab, right? A right and left lobe, a quadrate, and a caudate lobe. And the quadrate is next to the gallbladder, and the caudate is next to the inferior vena cava. There are ligaments, 5 ligaments. The falciform is usually evident on your models in lab. There's a ligamentum teres and hepatis, which are remnants of your umbilical cord. There's a ligamentum coronary and 2 lateral. So these again are just basic landmarks. I've never asked them in the lab. They're not shown on all the models that we have either. There is a lesser omentum. This is part of the mesenteries that sit over the organs, and so there's a lesser and a greater omentum. The lesser omentum tethers the liver to the stomach, so that holds them together. All right. So within the liver, there's lots of interesting pathology. We have a model in lab for this. Again, I don't really spend a lot of time on this in digestive, simply because the liver really only produces bile for us in the digestive function. So the lobes, if you look at them microscopically, you see these lobules, which are 6-sided structures, and within each one, if we magnify it, are these hepatic cords, which are a bunch, again, the cells, hepatocytes. There's always a central venule, lots and lots and lots of blood, right? the bile canaliculi, so small canals, right? So bile is produced by these hepatocytes and moves into these canaliculi, and that flows into ductiles, right? So, and from the ductiles, they go to a bile duct, which is at the corner, so six-sided, six corners, at the corner with a venule and an arteriole, and that's why they call that the portal triad. Again, that's all three of them together, triad portal for the type of circulation going on here. Sinusoids actually separate, so these are large spaces in the capillaries which allow arterial and venous blood to mix here. And we find, of course, macrophages all over the place because of these large openings, letting a lot of things move in and out of the blood. So I say all blood from the alimentary canal passes through the liver, and the liver will primarily process the nutrients and toxins from our diets. All right, for digestion, it's bile, and that aids fat digestion. All right, so remember that lipids are hydrophobic. They don't like to be with water. Well, all the secretions of your digestive tract are water-based, so you have a problem here. You have an oil slick on top of your contents. So the presence of fats in that chyme are not mixing with the rest of the chyme. So in order to help you digest chemically those fats and break them down, we have to basically get them to interact with the water-based stuff. And we call this process emulsification. This is not, again, a chemical digestion. It's a mechanical or physical. So you will take this large oil slick and break it into these big fat droplets. And this is done by bile. Bile is what emulsifies. So bile has a lot of different things in it, including the chemical breakdown products of hemoglobin, right, bilirubin and biliverdin, and they give that color to bile. It is alkaline because we're going to be dumping this in the duodenum, right, and the duodenum is an alkaline in nature. Bile salts are going to help with the process of emulsification. There's cholesterol, there's triglycerides, phospholipids, and electrolytes in bile. So it's a very complex mixture that we just refer to as bile. All right, so the bile salts and phospholipids are what really help us emulsify because they have both the polar and non-polar chemical regions. And so they interact with lipids and waters, and they're going to help allow them to interact together. So production of bile will increase when fatty chyme enters the duodenum. And that fatty chyme entering the duodenum will signal the release of cholecystokinin, and cholecystokinin will target the liver and gallbladder. All right? So the other accessory organ that's very important for digestion is the pancreas. I mean, really, almost all your major enzymes are coming from this structure. And it's an exocrine with a little bit of endocrine, as we talked about in the endocrine chapter, shown here is the pancreatic island. So it produces, again, a pancreatic juice, which is water-based, lots of salts. Sodium bicarbonate, huge, right, because we are neutralizing that chyme. So here, the stomach would be sitting right here, and it's putting all this really pH of about 2 into this tube that's not going to be able to handle a pH of 2. So the pancreas is making and dumping a lot of bicarbonate into the duodenum, right? The pancreas is making a bunch of enzymes to help with the digestion of proteins, fats, and carbohydrates, and even nucleic acids, because we do break them down as well chemically. All right. So when we go through each of those enzymes, which I think I'll save for the next video, you want to be thinking about this. You know, we have to get these chemical substances in your food small enough to enter your body. And the pancreas is going to be responsible for making a whole lot of different enzymes that allow that to happen.